Good morning, everyone. It's an honor for me to start this excellent day of the technologies and the presentation of the services that are available in our facilities here. I will start by reviewing what we are doing in the cryo-electron microscopic facility. So our facility works in both service mode and user mode, meaning that we can either provide the access to the instrumentation, data acquisition and analysis as a service carried out by my colleagues in the facility, or our users can get trained and can carry out their experiments on their own. So here is a brief distribution of our user base. Overall, it's more than 50 people nowadays. The dominant or the majority of users are internal people coming from the Brno region. And from those, the situation during the last year has slightly changed. We originally trained most of those people, so it was 70% of those who wanted to carry out an independent research on the instrumentation that we have. Nowadays, roughly half of the people who want to do the work on their own, and the other half who wants to take advantage of the service carried out by the facility staff. And also there is a portion of the people who come out from external academic institutions outside Brno region that primarily use the services, although there are exceptions which we allow to operate instruments on their own. And here is what we offer as a services, what we generally do. It is a standard set of things you can do with in the life science electron microscopy, from a simple imaging into a more complicated workflows such as lamella preparation and cryofit lamella preparation and cryo-electron tomography. So with this slide, we could actually end, because you now know all the information. But since that there is still some time left, I maybe go through a couple of workflows that we offer and try to show you why we purchase new tools and why we try to implement them into these workflows in order to make your life hopefully easier and more efficient. So the most utilized service that we provide and have is the so-called single particle analysis. This is a standard workflow in cryo-electron microscopy nowadays to determine high resolution structure of in vitro purified macromolecular complexes. For that, we need a device called the vitrification robot to plunge freeze your sample. Then we screen this sample in our microscope, one of the CryoEMs. And ideally, if the sample is good, we transfer it to the Highend TEM for the data collection. So since those people who kind of come around and see that, they will see that here and there that there is a new instrument, and also our colleagues from other facilities see that we purchase quite some tools over last years. So why we do that? And here I want to show that it should make a certain sense. So we were recently bought a second vitrification device so that we can have a substitution and a slightly different technology for our users when our original plunge freezer is down. In order not to prevent our users preparing their sample for the TEM. More importantly, we have recently adapted and optimized a workflow for preparation of the TEM grids coated with a monolayer of graphene, a mono or bilayer of graphene. And this has been optimized by Matthias Pinkas and is now available as a kind of standard type of a grid for our users. Why we do that? Monolayer of graphene is a monoatomic layer that does add a minimal noise, additional noise to the data when you collect over this graphene, and it is also suitable where if you have, for instance, complexes that take preferred orientation in the eyes, or when the user does not have sufficiently high sample quantity or concentration. However, utilization of this technique would not be possible without having a plasma cleaner for activation of the graphene surface that can operate with a light atom plasma. So we now have this Gatan Solaris that can operate with hydrogen, argon, and oxygen plasma. And on the other side of the workflow, we have purchased a computational cluster for our users that is available to all of the users from of our facility nowadays. It is 14 nodes of each node has 4 GPU cards. So it's a GPU cluster, and some of our users are already using it. For the rest, it's on my side. I need to finish the documentation or help and support documentation for that, and I will notify you in a near future how to get an access, how to carry on. We are undergoing a sort of beta testing with some of the users already. To add to that, earlier we also supported optimization of the sample purification, but we are discontinuing this as a service. So optimization of the sample purification for single particle cryo-EM to be more specific. All right, the other workflow that has been established here since the very beginning is the cryo-electron tomography workflow that is based either on plunge freezing of the sample and then directly collecting cryo-electron tomography data, or in a more complicated way, you then transfer your sample into this cryo-FIB-SEM instrument in order to observe the cellular samples and use the cryofib to cut or mill a thin lamella from your cell for the in situ structural biology studies, and then go back to the cryo-TEM for cryo-electron tomography. So for this workflow, we've expanded on different sides, or expanding on both sides. So for the sample prep, of course, there is a new plunge freezer, but not important. More importantly, last year we've purchased a high pressure freezer, which will allow our users to look at the bulkier specimen, look at the larger specimen, because it allows us to vitrify samples up to roughly 200 micrometer thickness. And since specifically for these bulkier specimen, one may want to then need to target and look inside with other means than inside a specimen with other means than the FIB. And for that, for 3 months now, we have this cryofluorescence microscope. So it is a wide field cryofluorescence microscope that shall allow you to target the fluorescent, this labeled organelles, for instance, inside the bulky specimen, which cannot be directly identified by the FIB. So we've expanded the workflow more towards the cellular specimen to be able to look at the largest samples, and also to invest time. We'll be now investing a time and effort to optimize this multimodal imaging of correlative light and electron microscopy.
Very soon, we should also expand the capacity on the cryo-FIPSEM side, because our current FIPSEM instrument is heavily used, and it's difficult to find a free slot, usually booked for a couple of weeks in advance. So we are getting a new dual beam system FIPSEM instrument. This time it will be a Highend tool. It will be plasma FIP that has also a capacity for high resolution SEM imaging, scanning electron microscopy imaging, and the plasma FIP with 4 different gases. It will enable a so-called cryo lift out, which is a technique to take small pieces of the volumes from this bulkier specimen. And it will also have an integrated fluorescent microscopy module. So for this part of the workflow, we will be probably able to also do the fluorescence imaging externally. And on the other end, which you cannot see here in the room, but you probably can see when you're online, we also, based on the request from our users, we purchased the software for the data analysis, which in this case is Amira from Thermo Fisher. Next thing is a new workflow that we are gradually establishing, because we did not have any suitable tools for that, and that is 3-dimensional studies of the larger objects, such as whole cells in a 3D, so-called which I here call an ultrastructural studies. This is a technique based on the embedding of the specimen into the resin, usually done by chemical fixation, dehydration, and then followed by embedding, and then followed by so-called FIPSEM imaging, or someone calls FIPSEM tomography, which someone also calls slice and view. For that, one may take advantage of having a high-end scanning electron microscope with immersion optics, which we do not have currently, but we were lucky that we got access for these initial studies to the Thermo Fisher factory, who provided us with the time on one of their instruments. And from that data, we can get an 3-dimensional reconstruction of the larger objects, such as whole eukaryotic cell at the ultrastructural level. And this workflow will be then brought in-house, or is being brought in-house, and will be further expanded. I've already mentioned the high pressure freezer, but then when it is combined with this freeze substitution unit, we can first freeze, or we can first freeze the sample at the near native conditions, and then basically transfer it during using multiple steps into the resin, as with the standard protocol. But this approach avoids and or diminishes the artifacts which are present during this sample preparation, such as extraction of the membranes. And then we have a choice to either go direct. We will have a choice to either go directly to the FIPSEM, because our new PFIP will have the capacity to collect high quality data, or we also nowadays have an ultra microtome that can be used to cut thin sections from that bulky specimen, and then carry out the TEM imaging. Yet another service that we have gradually established from scratch here is the electron diffraction tomography workflow, or you may also know it under the term of micro-ED, which is very, very popular nowadays. That is based on that is a tomography on the small crystalline material. In this case, this is paracetamol. We're primarily focusing on implementing this workflow for small molecules. and less focusing on the protein crystals of proteins. Well, we have, or Michal Babiak from the facility developed and software tool or piece of software application that can then ease the collection of such data, and also created a pipeline for processing of the electron diffraction tomography data. Of course, we're primarily focusing on the life science services, but since on the campus there is also other type of research. Some of our tools can be used for the research, for instance, in chemistry, such as nanoparticle characterization. And for that, we now have a new transmission electron microscope that is capable of scanning transmission electron microscopy data. So instead of taking a single image by flat beam, you scan across the surface and collect the data in a transmission. And this microscope has also the energy dispersive X-ray spectroscopy detector, which then allows for elemental mapping, so that our users can identify, for instance, the composition of their in this case nanoparticles. Further, I mean the plasma cleaner that we've purchased allows direct plasma cleaning of the specimen in the TEM holder, which is the routine technique or standard approach in the material science to clean up not only the sample but also the holder before insertion into the microscope, and that is also now available here. All right, so for the rules and the training, our active users probably know that they can find the laboratory rules in the sample prep laboratory, and that they also can find the standard operating procedures for most of the equipment that is there. If they want to refresh their mind on their own and to take advantage of the facility stuff, the current issue, and that comes directly to my head, is that it's only in a written form. We currently don't have electronic versions, and I'm very sorry for that. As for the training, I mentioned we train our users for independent operation. So we're participating in 2 courses at the Faculty of Science, one in the fall term, one in 1 year spring term, and we do the practical trainings on individual instruments, which are done individually in a group of 1 or 2 people. The trainings on the transmission electron microscopes are usually carried out by Michal Babiak and Karol Škudník. Then Jana Moravcová is now doing a training on the FIPSEM or cryofluorescence microscope. Radka Holbová will train our users on the high pressure freezing, free substitution, or the ultramicrotomy. And Vítek Koukal is focusing on the training on the STEM scanning transmission and the ES. So where we want to head in the near future, since I'm running out of time, I'll just go through that quickly. I mean, we've in recent years we've purchased a lot of or several tools to fill in the workflows that we want to provide to our users. Now I believe we should deepen our expertise in utilization of these workflows and advance in how we or in what complexity which complexity we can provide these workflows. One of such things is automation of the cellular preparation by CryoFit, and that is again a workflow piece of a software or application that Michal Babiak is currently working on, and which shall allow our users to run unsupervised cryo-fit milling sessions. Hopefully, we are now in the testing phase of that, and I hope that by the end of this year, I'll be able to present that or pass that to our users so that they can start also milling their samples overnight. So the other part is about the data. We see that there is a need to automate the whole procedure of transfer the data from the instrument to the final storage, and also find it very important to annotate the data in real time. And for that, Radek Veverka is developing a web tool that will allow you to submit the transfer of the data to the cloud directly from the microscope. It also allow you to start the data pre-processing or data analysis from the microscope, so you will become independent on the facility staff, which I believe is very convenient. It will be all operated through the web browser, and we are gradually working towards our in-house LIMS system, which I, however, will not touch on now. And also, of course, the last year told us that we should go remote as much as possible. So we're now thinking about ways how to implement a secure remote microscope operation for our users, and that is one other thing to come. Most importantly, you probably know that there is a lot of noise around here, and that is primarily due to us. So the facility will undergo an expansion, and from the current space that we have now, we will have more space in order to accommodate the new instrumentation for these workflows that I've just presented. The major benefits of the new space should be that there will be an operator room, so you will not need to sit directly by the microscope if you don't want to operate it remotely, of course. We will have a cell culture lab directly next to our sample prep lab, and there will be dedicated rooms with low humidity for preparation of specimens or this cryofluorescent microscopy under the convenient conditions. So these are the people who actually work for you. You probably know most of them, or you definitely know most of them. I'll just point out that we have a new person that's not in the picture. That's Daniel Pinkas, who joined us in September, and we will have Pelin Sungur joining us in February. Both primarily focusing on the supporting the TEM users. And we will also cannot see Radek Veverka, who is developing this web-based tool. And with that, I want to thank you for your attention. I'm ready to take the question.
Okay, so we don't have questions in the chat so far. So the floor is yours for your questions.
So far, so good.
I have a question about the breakdown application. The expected throughput.
So I mean, yeah, we don't do that blindly. We had a chance for a couple of demos in the factory here in Brno. Both myself and Jana were able to lift out and polish 4 to 5 lamellas a day in a kind of 8-hour day shift.
Is there a plan to partly automate
it? It is being worked on the Thermofish side. I know that there will be software that will automate, because it is a standard technique in the material science, where they mill the lamellae and lift out the lamellae, so that they try to utilize the same sort of software also for the life science business. So there will be something from them. Jana has tested it. I personally have not tested that yet, so I can't report on the throughput of this approach currently.
relevant. We have a few more minutes for questions before the NMR facility will.
Okay, so just for a case of curiosity, you mentioned this development on the software for the remote operation. I assume that, or yeah, for this. So I assume this is something that also other facilities around the world are focusing on because of the situation. Is your software like built up from scratch, or is it based on some already developed routines?
Okay, yeah, so I probably misspoke myself. So there are two things. One is the tools for the submission of the data to the cloud and automated annotation of the data. So adding additional information about the sample, and that is built by us. Because we have not found a convenient way how to use already available tools. And the remote access is something something different, where we will be using a commercial software for remotely accessing the microscopes. It is only a matter of setting a proper security levels for the users and authentication, the way of authentication to the microscope.
Okay, thank you.